Sorry, I'm muted. I'm stupid. I do this all the time. Anyway, hi everybody. Thanks for coming. I see you, Austin. I see what you got going on there. Do we need to reschedule? If we do need to reschedule, that's fine. We can. It's whatever. We can still make something out of this. I've planned for at least the next hour, so there it is. But and for those of you that have just sat through one show of mine and are now back for another, I really appreciate supporting the channel in that way. I know I'm taking up a lot of time today, but I wanted to jump on the opportunity to do this whenever as soon as we could, because it's an important this is really important science that precludes the creationist position. Like, not both of these things can't be true. So that's why we want to take a close look at ERVs and learn a little bit more about them and stuff. And I know Austin has been working hard on the subject, and so I look forward to hearing what he has to say. But if something's up and you don't think you're going to be able to make it here pretty soon, then that's fine. I'll talk about something else and we'll reschedule. We just ended the political show. Robin and I did. Had a great show there. Had a lot of people show up. Great turnout for it. Thank you everybody for that. If you're in for that kind of thing, we'll do it again next Thursday at 1 p.m. So that's going on. For those of you that didn't already hear all this, Saturday, the Skeptic will be here, and we're going to have a conversation about various things. He attended the art protest and what's up, New York City, and he has been making it to a lot of these events and stuff, and does a lot of videos covering creationist claims. And in fact, we have one particular creationist in common that I don't think he's aware of. Austin, you're fine. I've talked to Austin. He's told me that he'd be a little bit late or whatever, and that's cool. I don't. I waited a couple minutes just to see what was going on, but it's fine. No big deal. I can ramble for a little bit longer. And if something happens and Austin gets held up longer than he expects, then we'll just end it and reschedule. No big deal. The creationist argument, as we know, gets destroyed regularly by various different bits of science. And one of the things that we see, especially like whenever we look at the science of that would have to take place for creationist claims to be true, is that none of it supports that. Now, they try to take and misuse science, like trying to carbon date volcanic rock and stuff. Like there's a lot of stupid things that the creationists try to do and then call it science, which it's not. And it's not because that the thing that they're saying provides evidence for a God. It's because the things that they point to, number one, don't uniquely point to the existence of a God, and number two, are commonly misrepresented. That's really a big problem. In fact, one of the things that I hear often, they destroy evolution every time. Yeah, that's what they say. And that's actually what I was getting exactly what I was getting ready to say. Evolutionists, I hate that word. But anyway, creationists, they have they like to co-opt things that don't first uniquely point to their position, and second are generally like, how can I make it fit this narrative? How can I make the science fit in with what it was? And I'll accept anything that supports my position, and anything that doesn't, I'm gonna just deny. And that's kind of the way that goes when talking science with creationists. I don't really like talking a lot of science with creationists, um, but we got presentations here. um, I imagine this is something Austin's added. We'll see what happens here in a second. He's probably getting ready. Um, Evolutionarians, yeah, yeah. Um, Creationists say a lot of silly things, and they're they don't see them as silly because they think that their religious beliefs are the best lens to look at the world through. But their religious beliefs tend to not explain anything, just make claims about the world. The positions that there he is. What's up, man?
Not much, not much. Just
How many
busy, busy
PhDs
with
you got? Somebody wants to know.
and what do you mean by PhDs? You mean fat, huge dick? One.
Yeah, yeah.
I don't have a PhD. I kind of skipped that and went straight to working in the field of bioinformatics, biostatistics, biometrics. I managed to get a job working on biometric data algorithms and stuff like that. Even though I don't have a PhD, I went back to school just because I want to learn more so I can teach STEM, possibly at a college level. And also, one of the benefits of that is I get to shame and out intellectual grifters and frauds on the internet by exposing the nonsense that they spew, and giving the information that they withhold from their audiences.
Perfect. So yeah. When it
I
comes to
am
this kind of stuff,
I'm over 60% of my way to a degree in biology. So I've almost got the sticker. I've almost got the piece of paper to put on my wall.
yeah,
So I'm
yeah,
getting there.
yeah. So Austin is a student, and Austin has shown a lot of interest in specific areas and stuff, as well as some other things. But like,
Evolution is one of my specialties. Flat Earth is one of my specialties.
come on now.
And anti-vaccine hysteria is another one
Oh
of my
gosh.
specialties. And
Well,
those I feel like are really big, important ones. I'm also pretty good at debunking a lot of pseudo medical shit. Like there's a lot of medical quackery. So those are like my 3 or 4 areas of expertise, and I can debunk a couple of other things a little bit too, like astrology and quantum universe and like all these spirit science. Like that's pretty low level stuff that a lot of people can debunk, but I can debunk that stuff too.
yeah, and really the purpose of today was that I know that Austin has been working on this on the subject of ERVs for, and if you don't understand, endogenous retroviruses is what I mean when I say ERVs. Austin's been studying this for a little bit, and we had talked a bit the other day whenever I had refused to interact with Donnie and Matt, and
Yeah.
um, you were telling me like, hey, yeah, this for the argument they're making, this is the thing that really cripples them, and it's becoming a more common argument, I think, in this conversation because it does it it absolutely precludes the claims of creationists. Like they,
It does in so many beautiful and elegant ways, and it's difficult to explain sometimes just how uh, comprehensively endogenous retroviruses alone counteract a
oh yeah,
massive suite of creationist claims, including the intelligent design claim. That's one of the things I like about it, is that they are based on random insertion, which means that they are a powerful counteraction of intelligent design because they insert randomly into the genome. So how
Yeah.
can you have a random design? I can't see any way to do it in a way that logically follows, except by question begging. You know what I mean?
Oh
Like,
yeah, no, for sure.
uh, randomness can't be a design. That doesn't make sense. Like,
No.
it's like trying to argue a square circle in a very sincere sense. But
Yeah.
I put together a little presentation because I'm doing a debate with Donnie and Matt on the big old modern day debate channel that
Oh, did
sometimes
you get it
has.
worked out?
Yep, yep.
Good
It
deal. Good
has
deal. I,
sometimes over 1,000 people watching. I hope that that means at least one freaking person in there is somebody who genuinely is curious about whether or not humans and apes and other primates share common ancestry and whether or not evolution is true and they may be wrong about it. I don't think Donnie and Matt have any chance of deciding they're wrong about evolution.
oh no, no.
I think that's true for a large amount of creationists, especially the ones who create creationism content. So it's kind of a result of the skewed nature of voluntary sampling bias. The people who are more likely to tell you they're creationists are, you know what I mean. It's like you're more likely to find out about them than you are about somebody who's just like living their life and not spewing random crazy nonsense to
Yeah,
everybody who will listen.
like online, I will allow like atheists to use terms like evolutionists and naturalists and things like terms that don't really mean anything.
They
Um,
basically mean everyone who's not a complete dingbat.
yeah, yeah. It's
Uh,
like
yeah.
by not
I can be a little
accepting.
harsh. I've become very, very cynical about creationism. I think
Yeah,
for in a lot of the
it'll do that to
times,
you.
a lot of the times it's just kind of goofy and harmless. It's just silly
Yeah,
wackos. You know, the dumbest kid in your high school, uh, just going off. They're
yeah.
really just venting about all the things they're angry about. It doesn't really have much to do with evolution, uh,
So,
in my
so
opinion. And it's like, gosh, I forget what I was saying. Sorry, you had something you were saying there, Joe?
well, just to explain why we're kind of doing this. So, um, and you're kind of part of this too. So you've been working on a list of various sciences that are supported
Yeah,
or that would have to be denied in order to deny evolution.
basically.
And one of the things with all these different scientific disciplines is that the model that we use for the theory of evolution,
Yeah,
it's not just that it works within evolutionary biology, it's that all of these other biological sciences and some others also have to be,
geology,
it fits, it's the puzzle piece that fits all of these things,
100%.
right? And so the more we can talk about all those various disciplines and the primary arguments from them that argue in support of things like evolution, I think the stronger that we become to oppose the pseudoscience and the outright lies that many creationists tell.
Sorry, my
No, you're
daughter. Go ahead.
fine. Yeah, you're good. Yeah, sorry I caught you at a bad
Oh
time.
no, no, you're fine.
If you have something else to add to that, or if you want to go ahead and just jump right into it, I'll let you.
So do we want to share my presentation? That's why I came in on two different channels. The This is can I share my screen on the other one?
Yeah, yeah, yeah. Go ahead. It should just allow you to do it. I shouldn't have to. since you brought it in like that, it should be able to just put up whatever you put up. And
Oh,
I'll make it the big screen as you're talking about it.
it's odd. It's not. Oh, here we go. Share screen. Boom. Oh, it won't let me share my PowerPoint screen. That's interesting.
So,
Let me see here. Let me see if going into the slideshow mode changes that. Sorry to the audience. Try again. Share. Oh, is it share media? Is that the way you would do it?
you might be able to do it that way. Um, it does have an option in there for slideshows, I believe.
Oh man, I think I froze it. My connection isn't great. What's going on? Oh yeah, it froze on. Here we go. Oh, okay. Oh, I think I would have to have downloaded the slides before we began the live stream to do the share media thing,
Oh yeah, because it wants the file
or
from
uploaded them rather.
the. You should be able to just like if you're looking at them on your screen, you should be able to just share your screen. And even if you make that
I
like
when I clicked share screen previously. Oh, oh, oh! It's just playing the video I recorded.
This went a lot shorter and a lot faster. All right, history of evolution. We start with speed.
so, but
That's
I mean,
that's it.
we can do that and then pause it as we go if we, you know.
Well, this was just a practice run I recorded to check, to check it out for the debate. But yeah, you know what? It works. Let's
I
go with
mean,
it.
or we can kill the sound. What the fuck? We can kill the sound if you want, and do it that way too. So it's up to you, however you want to do it.
Actually, hold on. That was a video. Let me stop that. I think I just clicked the wrong file. Maybe I can share my slides.
Yeah, you should do it from there. Would be the easier way.
Share media. I clicked it again. It's taken a while because my
Yeah,
computer's
yeah.
slow. Yeah, da 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 da. How's
EV Mux
everybody
isn't
doing?
the
Do we
greatest
have a
on
good day?
the
Yeah,
desktop.
yeah. It's not bad. It's not bad. You know, it's tough when you're not like one of these super wealthy streamers that can put all this money into their process because people are sending them $100 super chats and stuff. Like,
You don't say.
yeah.
Well, you
Okay,
know,
so I see files, but I don't see uh PowerPoint files available in the list.
it seems that atheists are just as interested in having someone stroke their emotions as anyone else.
Yeah. Oh, here we go. Wait, no, that's the other one, isn't it? Yeah, it's not showing my slides. How odd. Share media. You would think if it can share the video, it would be able to share just the slides. Strange. Okay.
So let me. I can put a slideshow app up here, but I don't know. Let me see if it gives you access to it if I do that. Hold on.
Yeah, no. Oh, you know what? I figured it out. I
Okay.
figured it out, Joe. Now we're really cooking with gas. Here we go. Boom. Can you show can you put up my the presentation?
Oh, wrong one. There we go. Remove this one.
Boom.
Well, I'll leave it. It don't matter.
All right, so we're talking today about human chimp primate ancestry and evolution. This is genetic and morphological evidence. We're mainly going to focus on the genetic evidence today. So we don't need to get much into the history of evolution because I think everybody here knows the basic history of evolution. It starts with species fixity versus change of species. Aristotle, Anaximander, Linnaeus, ancient people arguing whether species are mutable or immutable. And that argument is pretty much over. Everybody accepts that species are mutable now. Even the creationists accept they're mutable, which is a really weird and often overlooked aspect of the evolution and creationism debate. Because the fact that they accept that species are mutable means that they're pretty much accepting there that the evidence fits the evolutionary model, which
and
is something that this is why it gets overlooked a lot, and why I think it is somewhat important to start off with the history of evolution a little bit. But we
just
don't have to focus on it.
hold on. Let me so I don't have to interrupt you. If I need your attention, I'll flash an emoji on screen or something. But when you say mutable, will you explain that?
Mutable, capable of change. So
Right.
mutability versus immutability just meant that if a species was immutable, that's the Kent Hovind claim. Hovind's claim is basically that species are immutable. Kinds produce kinds, Joe. After their kind, they will produce after their kind. Dog
You
will
got
produce a dog.
36
Whale
cows
will
here.
produce a whale. Yeah, exactly. Yes, that no, no, that is mutability. Saying that dogs all have common ancestry is mutability, because
Well,
that's saying that species can change. Hovind doesn't think that a dog has ever produced a non-dog, which is hilarious because dogs produced all kinds of dogs, as he himself admits. Again, this is part of why people debated mutability versus immutability of species even before evolutionary theory was a thing. Was because you could look and see that there are many types of dogs, there are many types of cows, there are many types of corn and tomatoes and squashes because people selectively bred them, which is just selection, non-random selection, acting on random mutation. That's another important part of bringing up the history, is because in this half of the eras, the part one half of the eras, we didn't know about non-random selection. A big part of the reason we didn't think evolution was as certain as we did, and we still debated immutability versus mutability back in this part of the timeline, was because we thought, oh, random mutation. We can see that there's variation and there's random mutations, but randomness can't create order. It can't
right.
create an ordered body plan. And then the second part of this era begins where Darwin and Wallace publish and figure out that you have natural selection, which is not random, paired with the random mutations, which are
Yes.
random. And that is capable of producing an ordered, new structured body plan and speciation and all of the things that created all of the biodiversity on the planet.
So
So,
that's the point that often gets made when you hear a creationist say something like that. Uh, I've totally blanked out there. Go ahead. Go ahead. I totally
no,
blanked out on what I was going to say.
no. And since today we're not focusing much on morphology, we can skip this slide, but just know that the gist of it is that basically everything a chimp has and is, you also have and is. You got the arms, head, legs, chest, pelvis, all in the same place, all the same shape, the bones, tendons, muscles, joints, your brain, heart, lungs, stomachs, intestines, gallbladder, testes, all that stuff in the exact same place. I think it's just mind blowing to note again, everywhere that a chimpanzee has a bone, tendon, muscle, or joint, you have that exact same bone, tendon, muscle, or joint. There's like one difference, and it's a bone some people don't have in their wrist and hand. Most people still have it. But anyways, the tool use, the emotion, yeah. The gist is basically everything that's the same. I'll take 1 second more on this slide to also add the final point, because this is an especially important and powerful point, and that is the vestigials and the tailbones. Humans have tailbones. We all know that. If you've ever busted your tailbone, it doesn't feel very good. But you'll live if you don't have one. But humans have them, even though we don't have tails. Most of us, at least. Sometimes humans are born with vestigial tails. This is something that happens in all kinds of organisms. They're born with vestigial features. It happens because of the way fetal development occurs. The way a fetus develops is it's got it starts off as a cluster of cells that divides and divides and divides until the tissues start specializing and becoming more specialized kinds of cells. And it does that by signaling for the cells to grow. And sometimes the cells start growing in a way that expresses the genome the way it would have been expressed in an ancestral form.
Yeah, these mutations can bring up things in our genetic code
Yes.
that are recessive and are no longer
100%.
expressed.
And that's precisely what an atavism is. And humans specifically are sometimes born with atavistic tail. Yes, somebody in the chat, native atheist with the win. Shallow Hal. Yes, this is an example that in the movie, one of the characters reveals at one point that it was one of these humans who was born with an atavistic tail, and they actually left his tail on, and it sometimes wags when he gets excited. They didn't depict the tail perfectly in the movie, but it's just a funny little thing. And yes, they do exist. I've seen pictures of them. I tried to find a picture to put in the presentation, but I didn't find any good ones because they're all babies, so they're all in some state of nudity. So, true freaking
Maybe not
video.
best
Can't look at the human body, dude. It's bad. But this is the important part of the presentation. The main thing we want to talk about: the genetic similarity between humans and apes, because this is the creationism killer of the evidence. There's just a ton of genetic similarity between humans and other primate species. And it starts off just at the base level with the fact that humans have 23 pairs of chromosomes, apes have 24 pairs, one off from each other. They have the exact same range of billions of genetic base pairs in their genome. Their genomes are all 3.1 to 3.3 billion base pairs long, so they're very close to the same length. And that makes them all very highly comparable. And then there's the actual numbers that we've produced by doing these comparisons. And I have a couple of those listed here. And that starts with the protein coding base pairs. The way your genome works, a lot of it, most of what it does, as far as we can tell, is just replicates itself.
Yeah.
There are other parts of your genome that do more than just replicate themselves. A lot of them, most of them primarily, produce amino acids, which are the building blocks, the monomer in the polymer unit that is called a protein. Monomer and polymer, those are just chemistry terms. You can think of a monomer as like one Lego block, and a polymer is like a stack of Lego blocks. That's basically all that really means. It just means the bigger unit is made up of the tinier ones. So most of your DNA replicates itself. Some of it produces amino acids, and even smaller amount of it does some other things, like it regulates the way other parts of the genome express themselves, so you aren't killed by mutation 100 times over.
And
And
and all of these things, historically and currently, I guess, are also controlled by the principles of evolution.
yes. So one of the ideas that I really like is that you don't really need anything but RNA and DNA and proteins, because once you have those, you have the blueprint that can make building blocks and structures. You have the building block and for making building blocks and structures, blueprints, the DNA, the building blocks, the protein. But proteins can also do a lot of other things. They can catalyze reactions. They can invert reactions. So those are a special category of proteins called enzymes. So with enzymes, proteins, and DNA, you can have basically a living system that's perfectly functional. It's just not held within a membrane. But then once you do have a membrane, that's pretty much a modern, like a protocell at the very least. Maybe even functionally not entirely
So,
unlike a cell.
are you familiar with the selfish gene?
Uh, Dawkins, gosh,
Well, I mean,
I hate
there's
to
other
endorse
people
him,
other
but it
than
is a
Dawkins.
good book. It is a good book, and I read it years ago, and it's spot on most everything it says. Things have updated a little bit on some other things. I mean, that's normal for a scientific publication. Nobody ever gets everything 100% right, but
Well, the only thing that I want to say about the selfish gene is the part where you said like it finds a something to move around in a membrane, and
yeah,
uh, there's a lot of talk about whether it's these things work together to further to make the individual the individuals within that population more fit to the environment, or is
Mhm.
it just the genes that this is the best way for them to reproduce?
Yeah,
Because sometimes sometimes it may be that those genes benefit from members of that population being killed off and are able to better reproduce themselves when they wouldn't normally without that be fit for the environment. So is it one or either or both that's driving that fitness? And when I say fitness, I mean
yeah,
fitness
yeah.
adaptation to your environment.
100%. Yeah. Sapolsky put that a really good way that I like. And it's a question you can ask yourself when you're looking at biological systems. And that is a chicken an egg's way to make another egg? Or is an egg a
Yes,
chicken's way to make another chicken?
exactly. Because you would kind of, if you look at early, at least theoretical early life,
Yeah.
then you might come to the conclusion that this goes all the way down to the individual genes themselves are
Yeah.
just trying to grow their population, whether it be within one individual or many individuals. So,
Absolutely.
the same rules will still apply all the way up.
And it reminds me of the original point I wanted to make. I lost it for a second, but that brought it back. Which was that um, you were talking about how the genes they can be selfish. You know, they can. It's the question of the chicken or the egg, right? And one of the things I like, and this isn't likely how we think it happened, but in abiogenesis studies, there's a statistical and chemistry based model of a 32-bit polypeptide chain, a 32-bit string of polypeptides, very simple, very small number of peptides, only 32. So theoretically very achievable. And all this polypeptide chain does is it replicates itself. But if you have even just that, theoretically, that is where evolution itself begins.
Yeah.
Because as it replicates itself, there'll be copy errors. It'll make, this thing just a little bit off or this thing just a little bit more right. And that is where natural selection will begin because some of those copy errors will do things like make it replicate slower and make it take more energy, more activation energy in chemical reactions necessary to achieve that replication. Or it might have a positive mutation, a positive error, where it does things like replicates faster or it requires less energy to
Or
achieve that
viruses.
replication. Yeah, or it replicates multiple times with the same amount of energy. Some crazy good, mutation like that, which is achievable in chemistry. Once you have something like that, that could be where evolution begins, and it shows how evolution happens at every level, both top down and bottom up. But that was a great tangent, and I really appreciated it. Let's get back onto the point a little
Yeah.
bit of genetic comparisons. The the telomere to telomere. This one is an important one to touch on real quick because a lot of creationists are misrepresenting this right now. There is a recent paper called You et al. That it completely sequenced the entire genome of multiple primate species and found that the intraspecies differences between primates, like if you compared a gorilla to a gorilla, are often close to or even less than the percentages of interspecies comparisons to primates. So, like comparing a human and a gorilla. And why this is a big deal is because if a creationist is going to try to argue that 86% similarity between humans and chimps isn't enough to establish common ancestry between them, they're basically essentially also arguing that 86% similarity between a human like me and another human like my own daughter standing over here making funny faces and distracting me, that isn't enough genetic similarity to demonstrate common ancestry between members of the same species that are known to have common ancestry. And the reason this is happening, the reason they're invoking this paper that destroys them so hard and telling such a silly lie about it, one that so deviously inverts reality, is because Casey Luskin told the clumsiest, most transparent lie about it ever. He cut out a graph. I think he did so using Microsoft Paint, literally. Uh, he cut
Yeah.
off the graph, which showed the intraspecies comparisons and just showed the interspecies one. Got caught red-handed by Dr. Dan, a friend of the channel. Love
Yeah.
Dr. Dan. He's awesome. One of his fans alerted him to it, and he caught the deception and sounded the alarm. And Luskin had to backpedal and go put in an appendage underneath the original post of the graph and add in the data he took out. Which I'll tell you one thing that might not have brought up. I think he did because he potentially committed something unlawful there by willfully misrepresenting that data and cutting off that part of the
Yeah.
graph. But of course, that is not what he mentioned when he did his appendage. He said, "Oh, I didn't include it because it wasn't relevant," even though it literally was completely
Well,
relevant. Yeah.
you're hitting on the point, which is the really the important reason to have a basic understanding of this, because um, a a number of creationists will use sources, and Donnie and Matt are extremely bad for this, to where they quote mine the hell out of
Yeah.
different scientific papers and just show the parts that support whatever
Yep.
claim they're making. And they've been caught red-handed doing this with ERVs and many other things. I made a whole video about them doing this with carbon or radiometric dating.
Yeah.
So that's why it's important to at least have a basic understanding of what we're talking about here, so that
There's a
because
great. Sorry. Go ahead.
Almost 100% of the time, it's real close to 100% of the time, when creationists give you a source, it almost
Yes.
always doesn't say the thing that they claim that it
There's
says.
a great catchy term for that, which is what I wanted to say. It's called a Trojan source.
Yeah, I like that.
Like the Trojan horse, the legend of the Spartans leaving the horse outside of Troy as a gift, and it's a trap because there's 100 angry Spartans inside.
But it would be, I think, though, if then they called it a horse, but really it was a cow. right? Like
Right,
that
yeah.
would be closer to the metaphor because it's really blatant. Like,
Yeah. So I'm going to do this a little bit out of order, and I need to go and reorganize this because I realized it should be ordered a different way. But the next point we're going to talk about is the second chromosomal fusions. So when we were talking about the basics of genetic comparisons, we mentioned that humans have 23 pairs of chromosomes and other primates have 24 pairs of chromosomes. And the thing I love about the second chromosomal fusion, and part of why it fucking honks me off so much, that Donnie tells so many freaking lies about this thing, is because it was an amazing, bold prediction by scientists that 100% paid off. They predicted, they did a simple calculation. 24 pairs is one more than 23. So if humans and apes shared common ancestors, that common ancestor probably had 24 chromosomes, we should see some signs of some sort of chromosomal deletion or fusion in the remaining pairs of chromosomes in our DNA. And that's
I agree.
exactly what we found. We
Yeah,
found centromeres inside of the telomeres that indicated that chromosome 2 was once chromosome 2 and 3.
yeah.
So we made a prediction. The prediction was verified. It's almost like the golden standard of science or something like that. Uh,
Novel
and
test predictions.
Exactly. And we did so using the same kind of genetic analysis techniques we use for criminal investigations, for genetic testing. So if this kind of analysis technique can tell you who is mommy and who's daddy, and who's brother and sister and aunt and sibling and so on, why in the crap does it suddenly become untrustable when it's telling us there's a fusion event in the second chromosome? And creationists have no other reason to say so other than they need to. It's a rescue device. Ironically, Donnie loves using the term rescue device for every piece of information there is about evolution. It's
Yeah,
really hilariously ironic when exactly everything he says about the second chromosomal fusion is nothing but an obvious, a painfully, bleedingly obvious rescue device.
yeah,
And one of the other great things about it, and I have this noted below, is we have observed chromosomal fusions happening. Cervus elaphus is the red deer, and Odocoilus virginianus is the white-tailed deer. And there was a chromosomal fusion in the latter species when they migrated to the Americas from the Old World. And we observed this chromosomal fusion happen. And obviously, it hasn't like killed any of the species off. They didn't become mutated or something. This is something creationists sometimes say about the second chromosomal fusion, that if the genome broke down enough for chromosomes to fuse, it would kill everybody off. The population would collapse, but that's actually not true.
no.
So, but then let's wind it back a little bit. We talked about uh, the genetic similarity and we talked about chromosome 2 fusion. Now let's talk about something that almost no creationists or evolution communicators, I feel like, ever talk about. And I don't know, I kind of know why. It's kind of a sub note and only really useful for this one specific thing. Cytochrome C. Have you ever heard of cytochrome C, Joe?
I can't say that I'm familiar with it. No.
Okay, and I figured you might not be because again, it is a little bit of esoterica. But the thing I love about this is that like the ERVs are already such a super strong argument on their own, right? But then what makes them even stronger is that there's a second argument that's almost equally as strong. It's even simpler and easier to understand, and the two correlate with each other. So they fit each other and support the same conclusion, like many lines of evidence supporting evolution do.
Sure.
And it is this which really makes this the Galileo moment for me. When I say that, what I mean is if you have studied history or the history of science at all, you're familiar with Galileo and Copernicus and the Copernican revolution, where we realized uh, geocentrism was untrue and heliocentrism was the truer model. And lots of people resisted it and rejected it. But eventually, under the weight of evidence, people had to relent and say, okay, this is proven. Yeah, the Earth rotates around the sun and not the other way around. Of course, except amongst the flat earthers on YouTube. Let's not even get into those freaking morons. Oh my good God. But this cytochrome C and ERVs are very much this for evolution, as if we didn't already have one with genetics. But it's always nice to have another one. And this one's like, it's even more slam dunk. It's like, the rescue devices are just insane. They're so stupid. Most of them don't even make sense. So this is what cytochrome C is. I have it listed up here on the slide if you want to take a second. If you're watching, one of the people watching this back, and pause it here and read this whole slide, you definitely should, because it describes it more in depth. But the basics of it are just what's on the first line and the last like 2 or 3 lines. And that is that cytochrome C is a gene, a sequence of amino acids, and it's highly conserved across species. Almost every species has cytochrome C, Joe. Fungi, plants, animals, protists, bacteria, so many organisms have cytochrome C. And the reason why is because cytochrome C is an essential part of how living organisms process nutrients. There's this thing called the Krebs cycle, or the tricarboxylic acid cycle. It's a cascade of chemical reactions that occurs in your microbiology, wherein your cells acquire their energy, their ATP, the chemical they use as their energy currency, through this cycle. And so, because cytochrome C is essential for that cycle, it's something that has been conserved across an enormous number of species. If you can't process nutrients, you starve and die. And so you don't pass on whatever mutation you had in that cytochrome C sequence that made you bad at processing nutrients. And so what's happened as a result is this cytochrome C is highly preserved across all the species. So many of them, so many species have cytochrome C, Joe. Let me tell you, all the species have cytochrome C. It's incredible. And my daughter likes my Trump impression. I'm trying to make her laugh right now. Anyways, all the species have cytochrome C, and what we've discovered with it, as we assess the amino acid sequences in certain species cytochrome C sequence, is they perfectly match taxonomic phylogeny. So what I mean when I say that, if you compare the amino acid differences between a chimp and a bonobo, there's 0. If you compare the amino acid differences between humans and chimps and bonobos, it's 0. The differences between humans, gorillas, orangutans, and rhesus, 1 difference, 1 amino acid difference. And as you can see, the amino acid differences in primate, monkey, all these different organisms, they don't get any higher than 3. Once you jump outside of the human relatives group and get into general mammals with things like badgers and kangaroos, you can have like 5, 7, 10 differences. You can see here kangaroos listed with 10 differences. When you get into some more of the distant parts of the kingdom Animalia, like elephants and birds, this big old megafauna creature has 15 amino acid differences between us. Birds have 10 to 12. But even something as different as yeast, a microscopic fungi, only has 44 out of 160 amino acid differences with us. The numbers are extremely small. It's extremely easy to understand, therefore, because the numbers are so small. And it shows such an immense amount of similarity because the numbers are so small. And those similarities perfectly fit the phylogeny that places humans as related to chimps, bonobos, gorillas, orangutans, rhesus, gibbons, spider monkeys, etc. So that's cytochrome C, okay? And then endogenous retroviruses basically do almost the exact same thing, just with a different meter. And the meter this time is viruses that infected a germline cell in your ancestors. So this is the candy. This is the icing on the cake. This is beautiful. Let me tell you, it's wonderful. So what is you probably remember from your biology classes, Joe, how a vaccine works, or not a vaccine, a virus. You remember the basics of how a virus works, right? Is Joe there? Oh, Joe's not there. Okay. Class, to remind you, if you don't remember, the way a virus works is that a virus does not have the tools within its own body plan to reproduce. It has to enslave a host cell and trick the host cell into producing the viral product for it by forcibly injecting its chromatin and genetic material into the host cell's nucleus, which tricks the host cell into uh, making copies of the viral product instead of the things that it needs for its fundamental life functions. So instead of producing its little packets of proteins and lipids and whatnot, it needs to energize itself. It's pumping out little copies of the virus. And what
So,
can sorry,
so a question about this, real quick.
yeah, what's up?
Now, whenever a virus injects its DNA, I guess, into a cell, does the virus then reproduce using the resources within the cell, or it just with every cell that reproduces, you get another copy of the virus? Like, does the virus eventually rupture from the cell and do something else?
What happens is the virus attaches to the cell. It uses all of the resources in the cell, all the resources the cell gathers while it's being controlled by the the virus parasite. I don't know why I keep saying vaccine instead of virus. The virus parasite, and basically the virus drives the cell to death until it's printed every copy of the virus that it possibly can, assuming it's the aggressive kind of virus.
Right.
Viruses are subject to natural selection too. So if you're too aggressive of a kind of virus and you destroy every host too quickly by so wildly and aggressively consuming its resources, you don't last very long because you don't spread. So they kind of, a lot of virus species strike kind of a balance where they consume the cell slowly enough that they can make a decent number of copies of themselves from it, or that some of them are capable of forming stasis with a cell to the point where they don't kill the cell at all. Maybe, and I'm not super familiar exactly with the entire minutia of that question, but it's a great question, and I think what I've given you as the gist there is pretty accurate. Does that kind of give you a better idea?
Yeah, no, it wasn't necessarily it wasn't anything that was
Viruses are
determined whether it was
they're
valuable or
very
not.
diverse.
Yeah,
So it would depend on the virus. Some
yeah,
viruses are so aggressive they'll rot your limbs off in a matter of weeks.
right.
You know, some are slow moving and barely noticeable even when they're in their higher stage, you know. But anyways, the ERVs, oh, this is relevant to both ERVs and your question, Joe. There's a specific enzyme that endogenous retroviruses produce called integrase. And it's the enzyme that they use to inject their genetic material into the host cell's genome. And the reason it's interesting to your question and the way ERVs work is because when you're looking at a genome, right? You're using a comparison tool on a computer, and it's a list of letters, A, T, C's, and G's, adenine, thymine, cytosines, and guanines. And when you look at the ERVs, what we have using these genetic survey data denoted as ERVs, you can see the sequences of base pairs that produce the integrase enzyme within the sections that are believed to be the ERVs. And they're believed to be the ERVs because I don't think I actually managed to list this down on any of my slides about them. Nope, I forgot to put that in there, so I should add it. The thing about ERVs is they have what's called long terminal repeats on either end of them. It's like a bookend. It's like a street light signaling everything after the first one is an ERV and everything before the last one. They're all the same endogenous retroviral insertion. They're all from one virus that injected its material into the germ cell of one of our ancestor species, individuals, and populations over time long ago, long enough that it eventually became fixated in our genome. And I'll tell you more about that when we get to this slide by telling you about the koala retrovirus event, which is an endogenizing event we witnessed happen. So we know this happens. So interestingly, not only do these, so that's the way the virus works. Sometimes the virus gets injected into a germline cell, like a sperm or an egg cell. And when that happens, that means all of the offspring of that individual are going to have the ERV in their genome. And it is through this process that it gradually becomes fixed in the genome. So when we look at what we think are ERV insertions in our genome and the ERV insertions of other species, what we see is that humans, chimps, bonobos, gorillas, siamungs, several primate species all share a huge percentage of them. The ones that we share most closely, chimps and bonobos, we share well over 99% of our endogenous retroviral insertions with them. And we share, again, much like with cytochrome C, an equal number of ERV insertions that fits the ratio of relationship by taxonomic phylogeny. So you share way more ERVs with a chimp than you do with like an elephant, but you still share a certain amount with an elephant, and you share even less with a fish and a bird, and so on. So much like the way cytochrome C works with the amino acids, the ERVs work with the genome itself, the genetic base pairs. And one of the cool things about it, and that makes it even more powerful, is the fact that once it has become fixated, it not only becomes a clue about common ancestry and speciation, it can tell us about when specific speciations occurred. So sometimes when you know how creationists are like, show me which one was the first human. You can use ERVs to get a pretty decent confidence interval answering that question, where you can be 95 to 99% sure the one is within the margin of error from a base that you've estimated.
So these germline insertions are they're events that take place within this population, and then we're able to then because it reproduces with the cell,
Yep.
that gets passed on in the germline through genetics
Yep.
to the descendants. So you can look and see that this event occurred, and it turns out like some of those events that may have been extremely detrimental to that population at the time now eventually lead to be very beneficial, right?
So I have heard varying opinions on that. Dr. Dan will say, yeah, there's kind of a decent amount of them that are useful. Larry Moran, who almost literally wrote the book on ERVs, he says no. There's
Okay.
like five. There's five that have been exapted to some use out of 200,000 insertions. And you know what? I'm kind of leaning towards Larry's opinion on this one. He made a
Oh,
very good case for it.
not
So,
well enough supported to make the claim anyway.
yeah.
Yeah.
But yes, so that is precisely it. And one of the things you can do because of that is you can do inferential statistics. You can do statistical modeling. You can take the number of base pairs and say there's this, because the number of base pairs tells you a sample space, a number of possible outcomes, you can estimate what the odds are of an ERV inserting in a loci where the denominator is the total number of base pairs in the genome. So 1 out of a billion or 1 out of 3 billion. And then you would be multiplying that proportion by itself for every common ERV insertion. So 1 over 3 billion to, gosh, like the 100th power, the dozenth power. It's once you're getting into that high of a number of exponents, the numbers are actually mind blowing. It doesn't look mind blowing when written in exponent form, but that's why we have exponent form, because the numbers are so mind blowingly huge you couldn't write them out in standard form.
Right.
And so what that tells us is, thanks to the way statistics works, the way estimating a population mean from a distribution of sample means in a Gaussian curve, which just means a normal curve, one that conforms to what's called the empirical rule. You can estimate these confidence intervals with like 99+% certainty. You can get well over 99%. I shouldn't say certainty, confidence. You can be extremely confident that the true mean lies somewhere between an interval you can calculate. And the level of confidence with which we're able to assess this is very high, not just because of that uh, statistical characteristic of these uh, common orthologous ERV insertions, but because there's 2 more points here, these last 2 points. We've reverse engineered some of these endogenous retroviruses. We took the the base pairs that were within the long terminal repeats, those bookending sequences that tell us, hey, everything inside of here is an ERV, and we actually reverse engineered the virus from the genes in our own genes, and it acted like a normal virus. It could infect other cells and get them to produce copies of itself. It had the structure of a virus. We literally took the scar this virus left on our genome and rebuilt the virus itself from that scar. And that's why, of course, we named it the Phoenix virus. That's one of the big points, is that it's really, really strong proof that that's what these actually are, and that our understanding of how these work is very confident based on our inferential statistics. We also have something called codon bias. And I understand these things, endogenous retroviruses, they are a little technical to understand. But once you understand them, it's mind-blowing how well they support evolution. And this one's a little technical too. I'm going to boil it down and really, really simplify it for you. The way protein printing works, and we talked about this a little bit earlier, is if in the genome you have 3 amino acids in a row, CTC, an AGA, an AAA, they produce an amino acid, they produce a protein. And the way that mammalian codon bias works, those 3 amino acid, or those 3 base pairs producing a protein, works completely different from the way the 3 base pairs producing a protein works in viruses. They also have codon bias, but it's a different kind. And when you look at our genome, you can see the mammalian codon bias in force for the vast majority of the genome, 92%. And guess what percent that part is? Do you think it's the part that's outside of those long terminal repeats? Or is it the part that's inside those long terminal repeats? You there? Oh, he's not there yet.
Sorry, I had myself muted. It's going to be inside.
It's the part that's outside is where the mammalian codon bias is. It's the part that indicates it's not an ERV. And then inside of the long terminal repeats, it is viral codon bias.
That's what I was. I answered you backwards because I thought.
Yeah, it's cool. It's cool. I figured it might be what was happening, but you get the point. Is that if you were to draw a number line that represented your genome and you put dashes on it that were long terminal repeats, inside all of the dashes would be amino acids that are base pairs, nucleotide base pairs that conform to viral codon bias, and outside of the dashes would be base pairs that conform to our mammalian codon bias. The evidence for ERVs, it's so mind-blowing. It truly is. And I hope you understood that part of my breakdown of it. And we're almost getting to the end of what I have here. This is just a couple of other points that just put it over the absolute just slam dunk that it is. And that is the koala endogenization event, the koala retrovirus. We are watching it become fixed in koala populations right now. We've been watching it for like 20 years, and over the course of that time, it has gone from being just an exogenous virus to being an exogenous and endogenizing virus to now it is fully endogenized and still exogenous. But over time, it will likely become just endogenized as the exogenous virus dies out. So we have watched the koalas endogenize a virus over, God, like I think about 10, 20 something years. And that is, again, just really powerful evidence. So we see the long terminal repeats and we saw it happen. And of course, this endogenization event also produced the long terminal repeats in the base pairs that the koala retrovirus is contained within. And then creationists might say, oh, this is secular science. The secular scientists are just lying. And that's hilarious, too, because most of this comes from Larry Moran and Graham Finley, who are both evangelical, devout Christians. Finley is even a preacher, in addition to being. So, no, they can't cry, secular conspiracy on this one. They're both devout Christians, but of course they accept evolution. And then the last thing is, so I said that they were just that that endogenous retroviruses were so strong because it's actually two arguments that fit each other, endogenous retroviruses and cytochrome C. But actually, it's like more than a dozen because there's also these thing called jumping genes in your genome that jump around from one insertion spot to another insertion spot, and they do this on the endogenized DNA, and they do this on cytochrome C, and we can mark those and track those. So there's already these really, really low odds of common insertion events, but then you also need all the common jump events, all the common mistranslation events where one jumped into an ERV segment of base pairs. And all of those are similar too. So you can liken it to like finding two bullets at two different crime scenes that are the exact same caliber and have the exact same congruent scratches on them. And what that tells an investigator is the bullets were both fired from the same gun, from the same firearm, and were scratched by the same common barrel, the same rifle. So in this case, our genome is the bullet. The ERVs and all of the common markers in them are the congruent scratches. And then the common gun that fired them is this common ancestor that we had somewhere in our line that had an endogenization event in one of its germline cells.
So it's some kind of like low level phylogeny.
Yeah. And so once you've like understand it, it's really simple to actually kind of intuitive, and you can see how it's really strong proof of macroevolution and common ancestry. Like we're literally talking about the common ancestor and what happened to it in that population's lifetime in terms of its virological record.
Well, I think obviously there's a lot of information to go over and in support of what you've laid out here.
Yeah,
And the idea of this was to show basically just like this is why this argument is a problem for creationists and a young Earth
absolutely.
and for arguments against evolution and all those things.
Yep.
And then now, if you're interested in this, it has to become up to you at some point to then go and find more information on it if you wish to understand it better. And that's what I'll be doing. I will share. Well, first of all, I've put Austin's link in the chat there, and you can go over to his channel, and he talks a lot about things, this and other things related to this. So you can get a lot of good information there. And as I look more into this, I will post sources on my Discord server. So if you're over there, that would. Oh. Did I just lose the light? Okay, yeah. So you can't see because I turned off the camera, but um, this will be something that I'll add some more to. And if you're looking for information, the first place I would send you is over to Austin's channel. Though, go over there and subscribe. He's got videos on lots of stuff like this.
Yeah, I even have a short on ERVs that explain what they are in under a minute. So share that around if you want to explain what ERVs are to somebody in a really quick, easy 1 minute video. And then one thing you forgot to mention there, Joe, is that they also destroy intelligent design. And that is what this last slide is about, because this is what is most likely going to be a creationist's reaction to endogenous retroviruses and cytochrome C and genetic similarity. Yeah, there's common design. It has a common designer. You think it has a common ancestor? I say it probably does if it's a cat compared to a cat, but not a human compared to a cat. It's common designer. Yeah, Hovind. Yeah, no. So this slide is just a series of questions about what all of the things that are necessary for ERVs to work the way they do are designed for, which of course creationists have no answers for. Bless their little hearts. They tried for at least one or two of these. I imagine a couple people have plugged it into an AI and been like, refute this, please, AI sycophant. And the AI sycophant was like, I really can't, but I'll do my best. And then, give them some stuff to spew out.
Yeah.
And but yes, these are some simple questions, and it's real simple. Just asking them what the specific parts are designed for. Why does it have the integrase enzyme in there? What was that designed for, if not for injecting the chromatin into the host cell? Why would that be designed into it? And yeah, they don't have any answers for them. And I know especially Donnie doesn't, because
Yeah.
Barry Desborough, John Peters, Dr. Dan, I'm sure like 4 or 5 other people who aren't as well known. I'm sorry if I forgot you. Maybe Dr. Chris Thompson too, I think. God, I need to get more familiar with Dr. Thompson stuff because he's awesome too. But they have all been hounding Donnie about this, and anytime any of them bring it up, there's a Donnie-shaped hole in the wall, like a
Yeah.
Looney Tune.
Well, my first experience with seeing Donnie address this was in a debate done on Standing for Truth with Mark Reed.
Yeah.
And Donnie tries to do his usual thing where he has these slides that he's labeled at the bottom that they all came from creationist sourced places, and then Mark Reed stops him dead in the middle of it and says, "Wait a minute, Donnie, I have that paper right here, and the paper you're talking about actually goes on to show how ERVs absolutely support the theory of
Yeah.
evolution and explain it perfectly. And you're trying to use one line from that very paper to say that it defeats evolution.
Yep.
And
More Trojan sources.
yeah, this is Donnie's game, and it always has been. I mean, this has been going on for some time. I look forward to. I'm sorry, I won't be watching you on modern day debates, but
That's
but
sweet.
I do look forward to finding out that
I
you.
am doing it mostly as a sociologist. I plan to go on, and I do want to ask before the debate begins for everyone in the audience. And I know this is terrible. It's skewed by the voluntary sampling bias. Ask everybody watching to tell me in the chat if they are somebody who's willing to change their mind about evolution. Like, is this debate a waste of time? Is anybody in this audience changing their mind? I don't think it is entirely a
I don't
waste
either.
of time, but I think the numbers might not be very high. But
I think
again,
that number one.
that's skewed by voluntary sampling bias. The people who are more likely to tell me are going to be the people who are like really strongly convinced of something.
I think that number one it is one of the terms that I use, and I don't use this much online, but when talking with friends about this kind of thing, is that it's easier to believe a lie than it is to go find out the truth.
It can be.
And I use this in my political talks and everything because I think that you're going to have a lot of people that it's just like this is a complex topic. When you really get into it on the surface, you've done a great job of explaining it in a way that I think anybody who's wanting to understand can.
Yeah,
But
but to be fair, it took like an hour and 20 minutes. I have to try
yeah,
and get it done in 16 in my debate with Donnie.
but you won't have me interrupting, and we
And yeah,
started.
I was gonna say yeah, and I'll go a lot faster.
We
I
got
got
some
the really fast debate pro talking shit down, no problem. I can talk really fast and really
yeah,
get yeah,
I have to do the opposite there because
yeah.
whenever when I write something that I'm going to say,
It's a
I
trade
have
off.
to
It
I
gets
have to
you
slow
through it
down.
faster, but people are less likely to absorb what you're
Yeah.
saying unless they too are really intelligent, and most of the really intelligent people are
Well,
already on our side, I think.
I
Gotta
kind of
slow down a bit.
learned that it's important to whenever you come to a point on whatever it is you're saying to
Yeah.
kind of at that point then
Pause.
pause and let that
Yeah,
sink in.
for
Yeah.
sure.
And I'm bad about just rush like this is what it is and this thing and this thing and this supports it and this doesn't and let's talk about this because that's a problem too and this is bad. And I I kind of get I'm bad about rushing through it like
Yeah.
that. So
Yeah. No,
I
I think
understand both
I
angles though.
think taking your time is valid. And that's one of the things that's really weak about one of debate's biggest weaknesses and why it's often more performance art than actual learning and education. But when debates are done well, they can be educational.
Well, I'm actually as far as like the debates that I take with creationists and stuff, I'm confident in the science and the truthiness of it to assist me quite a bit. And right now, the reason that I'm doing a lot of these is to be able to talk about those things better.
Yeah.
Um,
No, that's fair.
my, yeah, to be able to express and articulate that stuff because I think the value of information has a lot to do with your ability to communicate it to others. Uh,
The debate is November 21st. Average Joe Cool, atheist. Thank you so much for being interested.
yes, and it's not that I may come watch. Actually,
No,
I won't say that I won't.
I totally understand. The channel is toxic and it's noxious, and I am really I considered not taking the debate, but
Yeah,
I thought about it and decided it was worth it just as a learning experience for me to maybe boost my channel a little bit and to get some possible answers about whether or not there are people in that audience that can be convinced. And if
Well,
there are, I will put more effort into convincing them. And if there aren't, I can fuck off and not waste my time.
yeah, my position with modern day today debates has a lot to do with. I know that some people see it as like a moral question to go watch it or interact and
Yeah.
all that stuff, and there's that, and I understand that, and that does influence this, but a lot of it has to do with my personal experience dealing with them when I did a debate there before. So
I've heard some bad things.
I've actually talked shit about modern day debates way before the thing happened with Matt and all that crap.
And,
Like,
and I've criticized them too. I've criticized them in the comments on their channel and said, James, you got to stop calling this an unbiased platform. It,
yeah,
it's not an unbiased platform.
it's a very exploitive platform, I think.
You're right. Yeah. Yeah.
But I'm willing to always to cheer on a friend or whatever in whatever endeavor. I don't. I'm not of the mindset, as some people are in this community, that you must agree with me on every topic and do what I do, or we can't talk and be friends, and I can't support you, and all that shit. I'm not into that.
Yeah. No, I'm a broadly humanistic rationalist.
Yeah,
I can try and love most everybody.
I draw
Even
lines,
though
but
I
that's just not where I'm at.
vehemently disagree with them. There are lines that I will draw to.
Yeah, yeah. So
And
and that's fine. And I respect each person's ability to do that. I think you're a smart, intelligent person, and I trust your ability to see the situation for what it is.
that's so polite and nice of you. I'm an idiot. Absolutely do not trust me on anything. I'm an absolute moron. Look up all this shit yourself and find out how I'm wrong about it and come tell me what a stupid dummy I am. And I can't wait to hear about it.
Yeah. And so I'll if you want to go over and tell Austin what a dummy he is, I will put his link in the chat again. And also, anytime you're here, all you got to do is top that right there, and it'll post your stuff, and you feel free to do it. I don't care. But So again, this was just a point for us to get to where we could talk about the basic parts of it, so that now I can go and look at specific things that you said and do a little more research myself, because that's how I'm going to learn it best.
I will send you some links to two blogs, one written by John Peters and one written by Barry Desborough, and both of them are drawing from the works of Graham Finlay and Larry Moran. Those four sources should give you everything you need.
Cool. And I will make sure that those are reposted in my Discord account for everybody that's over there.
Yeah, I might not get to them right now because of busyness, but I will eventually get them to you.
I know you just got done with stuff and just picked up your daughter, so we'll kind of wrap this up here so you can
Yep.
get back
If
to
we
your
get,
life.
yep, that's what I'm signaling is we're getting to the end here. If I can give the audience a final point, is you can keep it simple. ERVs are common viral insertions in your DNA that are in there because we share a common ancestor with the other species that have them. And you can be extremely statistically confident that we are not wrong about that. Like outrageously statistically confident, like well over 99% confident. So if you can be 99% confident in that, you can definitely say that evolution has evidence and is well supported and all that fun stuff. And you don't creationism, intelligent design, species fixity only has 0.00001% confidence. And that is what makes ERV so powerful, why they're a new Galileo moment. And that would be the one main point I hope you learned from this. And
Well,
learned it well enough to explain it to other people.
Jester has an important point here with the $2 super chat. You're a big dumb doo doo head. Oh wait, that's me. Thanks for the $2 super chat.
You're so sweet.
Jester's always good with
Oh man, we love Jester.
not only is it awesome that Jester sends in the super chat, but the message that comes with it is always just perfect. So
Right.
yes, but I will get everybody out of here. You've seen my face for 2 different streams today. I'm sure
Yeah.
you've had enough.
You're tired too.
And Austin, I appreciate you coming and helping me learn a little bit here so that I know which direction to go with this. And everybody go over to Austin's channel and subscribe, please, because he definitely deserves your subscription. Yeah, so good luck on the debate. You might hear me bring this topic up in a future debate or something too. So
Absolutely.
I'm going to wait till I get a little better grasp on it. But I know at least you answered some questions and things, some basic questions that I had, and so I know at least which direction to go. Do you got anything else you want to plug or say before we cut out of here?
Just that if you know the basics, you can express that you are pretty confident you're not wrong. You don't have to convince everybody else that it's true, but it gives you a good, strong indicator that you're not wrong.
Yeah.
And I'm sure you'll hear all kinds of nonsense, Joe, if you do bring that argument up, but that's a good thing to remember.
Yeah. And that's one of the things with a lot of things that I do talk about is I'm confident that the facts are on my side of that thing whenever I'm doing it, and that's what I look for is the integrity of the thing that I'm putting forward as information to everybody, and I think that this has that. So
100%.
definitely.
Yeah. You got to remember we're all human. So if your epistemology doesn't count for the fallibility of your own senses, it's probably poopy.
Yeah. Looks like you already got a couple new subscribers over there.
Oh,
Thank you guys so much for subscribing to Austin's channel. And I know you've already clicked the like button on this video. So thanks for watching. Thank you to my channel members. Thank you, Jester, for the super chat. And as Moondog there says, orange people suck. Thanks for coming, guys. Have a good one.